Alright, listen up. If you're planning on sitting the 2026 A2 Cambridge Biology exam, you need to stop what you're doing for a second. We aren't just looking at a syllabus. We are looking at a blueprint for success. Today, we're deconstructing the 5 must-know topics of the 9700 syllabus. If you walk into that exam hall without mastering these, you're essentially leaving your grade to chance. Let's make sure that doesn't happen. Before we move on, let's talk about your final prep. You can know the theory, but the exam is a different beast. At Blackwood Prep, we've engineered our 2026 A-level predicted papers to bridge that gap. These aren't just practice questions. They are data-driven simulations of the actual papers you're going to sit. We've included the exact unfamiliar contexts and tricky synoptic links that catch students out every year. If you want to walk into that exam hall with 100% confidence, grab the predicted papers at the link in the description. Invest in your grade. Now, back to the lesson. First, let's talk about what the examiners actually want from you. Think of these as the 3 pillars of your revision. First, absolute precision. Using vague language like breaks down is a one-way ticket to losing marks. You need to say catalyzes the hydrolysis of. Second, unfamiliar contexts. They will throw a weird disease or a plant you've never heard of at you. Don't panic. They're just testing if you can apply core mechanisms to new data. And finally, the synoptic mindset. Biology isn't just isolated chapters. You need to link the theory in paper 4 with the practical analysis in paper 5. Topic 1 is genetics. Think of this as the probability engine of life. Everyone knows monohybrid crosses, but for a grade A, you've got to go deeper. You need to confidently map epistasis. That's when one gene locus masks another, like in feather color for chickens. And don't forget the core engine of variation, meiosis 1. It's all about independent assortment and those crucial swaps at the chiasma. If you can deduce ratios beyond the standard 9:3, 3:1, you're ahead of the curve. Let's zoom in on the lac operon. This is a classic on/off switch. In state 0, the repressor is parked right on the operator, physically blocking RNA polymerase. But when lactose shows up, it binds to that repressor, changes its shape, and it detaches. Suddenly, the path is clear, and transcription for $beta-galactosidase proceeds. Pro tip, never confuse the promoter, where the polymerase binds, with the operator, where the repressor sits. Topic 2 is evolution, the drivers of change. You need to distinguish between continuous variation, like height, and discontinuous variation, like blood groups. But the real meat is in the selection pressures. Are we talking stabilizing selection, keeping things average, directional, shifting toward a new extreme, or disruptive, favoring both extremes? And if you want that grade A edge, mention genetic drift. Sometimes evolution isn't about the fittest. Sometimes it's just pure chance in a small population. Math time, but don't let it scare you. The Hardy-Weinberg principle is your framework for population genetics. P plus Q equals 1 for alleles. P squared plus 2PQ plus Q squared equals 1 for genotypes. Here's the trap. This only works in a perfect world with no mutations and random mating. When you're calculating, always start by identifying Q squared, the observable homozygous recessive phenotype. Work backwards from there. Topic 3. Engineering life. This is the how-to guide for modern biology. You isolate the gene, amplify it with PCR, insert it into a vector, and then identify the successful clones using markers. The grade A secret here? Explain the reverse transcription pathway. Using mRNA to make cDNA allows you to skip those pesky non-coding introns that bacteria can't handle. We also have to look at the cutting edge, CRISPR-Cas9. It uses guide RNA to find a specific DNA sequence and snips it with the Cas9 enzyme. It's incredibly precise, but compare that to traditional gene therapy. While gene therapy offers hope for disorders like cystic fibrosis, it faces huge hurdles, immune rejection and the ethical minefield of germline editing. Topic 4. Photosynthesis. Think of it as a 2-stage engine. In the thylakoid, light energy splits water, photolysis, to create ATP and reduced NADP. Then, in the stroma, the Calvin cycle uses those products to turn CO2 into sugar. Remember, 5/6 of your triose phosphate goes back to regenerate RUBP. Only 1/6 actually becomes the sugars and lipids the plant needs. But the engine can only go as fast as its slowest part. That's the law of limiting factors. If light intensity is high but the rate of photosynthesis flatlines, then CO2 concentration or temperature has become your bottleneck. We also use chromatography to identify pigments. Calculate your RF values by dividing the distance the pigment moved by the distance the solvent moved. It's a definitive ID every time. Topic 5. Respiration. It's a 4-step staircase. Glycolysis in the cytoplasm, the link reaction and Krebs cycle in the mitochondrial matrix, and finally, oxidative phosphorylation on the inner membrane. This final stage is the big winner for energy. It harnesses a proton gradient to generate a massive yield of ATP. Oxygen is just the, quote, final electron acceptor that keeps the whole chain moving. If oxygen disappears, the whole system stalls. You're left with just glycolysis and a measly 2 ATP. You can actually figure out what an organism is eating by looking at its respiratory quotient, RQ. Carbohydrates give you a clean 1.0, while lipids are down at 0.7. If that RQ goes above 1.0, that organism is doing a mix of both aerobic and anaerobic respiration at once. Now let's talk about the practical hurdle, paper 5. You have to plan the protocol, analyze the data, and, most importantly, critically evaluate. Here's a huge tip. Human error scores 0 marks. Don't say it. Instead, identify specific methodological flaws like colorimeter fluctuations or evaporation from unsealed tubes. Give the examiners concrete improvements. Think you've got the right answer, but still losing marks? You're likely falling into one of these 3 common traps. Let's fix that. Trap 1. Being vague with data. Don't just say the graph goes up. Use the numbers. A grade A answer quotes the data. The rate increases from 5 to 25 units as temperature rises from 10 to 40 degrees. Always back up your claims with evidence. Trap 2. Basic verbs. Stop saying enzymes break things down. Use the technical mechanism instead. It's better to say, the enzyme catalyzes hydrolysis by lowering the activation energy. Specific verbs win marks. Trap 3. Loose vocabulary. Don't just blame a mutated gene. Be precise. It's a mutated allele that results in a non-functional polypeptide sequence. The difference between a C and an A isn't just knowing the facts. It's using the right language. Stay sharp. That is your 2026 A2 Biology Blueprint. We've deconstructed the syllabus and looked at the exact mechanisms you need to master. Biology is tough, but with the right strategy and the right resources, that A-tur is completely within your reach. If you want more deep dives like this, make sure to subscribe and check out the predicted papers at the link below to start your practice. Good luck with the study sessions, keep pushing, and I'll see you in the next video.